Depends on how warm it gets between now and the end of. No, just.
I barely.
Yeah.
It's so embarrassed. This is something.
Oh, that's awesome. It sounds like you will, have some pretty good chances.
Yeah, that's why
Hi.
I felt everyone's good. A lot, but I still, I think I have a good chance.
Do you do any photography? They're doing a photo contest too.
Oh, really?
Yeah, I think you get $100 if you win, in one of the 6 categories.
You're gonna do that?
Uh-huh, why not?
Like a bet?
Huh? So
Okay, so let me just make sure about the field trip. I know for sure that Beverly and the Jessica are writing. And Sock is thinking about it, and you're gonna meet us there.
I will not be.
Oh, you won't be there.
Yeah.
Okay, but you two will be there. Okay, try to get there by around 10:45 if you can. If we make good time, we'll get there. And I just wanna make sure that you're there when I check in with other people, so I don't have to wait 10-15 minutes. for
what
you.
time are you guys leaving from here?
We're gonna leave at 8. And we have a car, we have a sedan. So that, we'll be able to go down faster than we did in the van. I don't like to drive that van at 80 miles an hour with students in it.
So I actually, so I have a committee meeting, right? Friday. next
Is it,
Yeah, so coming up Friday.
yeah, that's not, we're going this
I know,
Saturday.
I know, but I need to finish like some. I need to submit some.
That's up to you, you know, what you need to get done, so don't let it interfere. These field trips are optional, they're not required, so. Okay,
You want us to meet here at about 7:50 then?
yeah, why don't we meet here, say 7:55, 5 minutes early, and then if everybody's here, we'll just take off. That just means more time that we'll have in the, at the exhibit. Okay, I posted, the student prospectus. And that'll be due a week from Friday, that's just before you leave, and it looks like this, okay? So this is kind of the next step. Last week, you told us what your project, the title and what it was. Repeat that project, briefly describe it. And then this is something, I actually got this from Rosemary Smith. This is what I think they use for their undergraduate research projects at Rocky Mountain Biological Labs. So project site and setting, where and whom will you work? Major questions you hope to answer or goals you hope to achieve. Provide 3 or more key references, okay? So you're not doing these things independent of anything else that's ever been done. So you might want to find, you know, a key reference might be the Gatorland website. Or maybe there's a really good video that, you know, you're trying to thing, or maybe a time lapse study or something like that that's been done. Okay, and then for those of you that are doing more scientific approaches, you know, like, it'll be easy for you guys to find the key references, right? Or some key references for that. Sock's already got his, so. Anyway, but I wanna make sure that you guys have gone into the literature and looked around and seen what else has been done on your project, okay, or on your area. What do you expect the results to be from your project? What are the products going to be? The resources needed. What do you need in order to do an excellent job? If there's something you need, let's get it identified now. If it's a book or it's a piece of equipment or it's some information from a company or something, let's try to get that taken care of before spring break. And then this is kind of the hardest, this is something that really makes things real, is make a calendar and say, break your project down and say, okay, by this time, I'm gonna have my references done, by this time, I'm, I'm gonna make sure I've got all my equipment, by this time, I'll have gathered the data and so forth, okay? And then watch you fall behind, it's like, and then, your biggest concern or questions about the project. What is it that bothers you the most about it? And then I can look at that and say, well, look, I know how to solve that, so. Okay, and if you wanna turn this in earlier and tell me about it, that's fine. If you wanna talk about it before we break for the springtime, okay? For the break. So that's that. Today, we're gonna do gas exchange. I've been finding some really interesting herp articles in the news, so I'm gonna show you some of those. One of the really interesting ones is they're developing synthetic antibodies for snake venom. And it may be possible for them to come up with a universal anti-venin for snake bite that'll work on any snakes. So that's a pretty exciting thing. And then Monday, I'll probably need some time to talk about spatial ecology. What I wanna know is, do you guys feel that, do you wanna have a review session for the test the next day?
Yeah.
Sure.
So that helps you. Okay, so we'll plan to do that. Okay,
I opened the gas exchange PowerPoint and it says water relations instead of gas exchange,
oh, did I put the wrong one up there?
probably.
And
Oh.
it says I have same problem.
Oh, well. Huh, I wonder how I. Hmm. Do you use that while I'm lecturing? Do you want me to try to fix it right now?
If you can, I will appreciate
But
it.
let me try to fix that.
Yeah, that's the next. I'm working on my thesis.
This is actually the PowerPoint. I haven't made up the PDF. So that's what you'll get. Try it now.
Thank you.
What I do is I make a mistake. I copy the previous one, so I don't have to set all the things, and then I forget to exchange the file. What should happen? Okay. So pull that down and tell me if the right
Yeah,
thing,
it worked.
does
Thank
it
you.
work? Okay, good. Sorry about that. Okay, so we've actually talked about quite a bit of this already. Why do animals need to exchange gases? What are the different gas exchange surfaces? So, gotta get oxygen, right, for aerobic respiration. What happens if you don't have oxygen? Besides dying, I mean, why do you die?
Because you don't have oxygen for your blood. It's not for your blood.
And why do you need oxygen at the cellular level? This is introductory biology.
Isn't it because the CO2 will build up and it turns your blood acidic and you die from that? Where's that?
Well, that's true, but that's not why you need oxygen.
Need it for cellular respiration.
ATP.
You need it for cellular respiration, you need it for the Krebs cycle, right? You can have anaerobic cellular respiration, but that's not enough to keep an animal like us going, keep a vertebrate going. They need much more efficient, and so it's required for the Krebs cycle to generate the ATP and the energy that we need for living, okay? And you need to get rid of the carbon dioxide, because, when you take a molecule like a sugar and break it down, it'll break down into carbon dioxide and water. Okay, what are the different gases? Would you guys not please talk during class?
No, we were talking about the answer.
Okay. What are the different gas exchange surfaces? Okay, so the non-pulmonary, these are the ones, not the lungs. So we can, put these into lungs and pulmonary and non-pulmonary. So non-pulmonary are gills, skin, the mouth, buccal cavity, and the throat, the pharynx, and the cloaca. And then the pulmonary ones are the lungs. Okay, we went over this last time. I actually, in your notes, well, actually, the ones you just downloaded now will be correct. But in the notes, I had not put surface area in there, but an extensive surface area really is an important property. So I added that to that. But the one that we didn't do when we were talking with the Menti is vascularization. Okay, it's really important that blood in a multicellular animal, like, like in an amphibian and reptile, that the blood gets into contact, gets close. And then you can use bulk flow of the blood to spread that oxygen out throughout the body. But if you had to wait for oxygen to diffuse from the skin, like into the, to the heart or something, that it would just take way too long. So vascularization, the blood, having the capillaries right there to exchange gases is really important. Okay, many amphibians and some reptiles use more than one respiratory surface. That's particularly true for amphibians. And that may be simultaneously, or, in development, it may be they switch from, gills to lungs. Or they rely more on skin when they're small, or an embryo, or a juvenile, and then less when they're an adult. Okay, so let's start out with non-pulmonary gas exchange. These are for all larval amphibians, will have, I think all larval amphibians will have gills. This is a tiger salamander, and you can see those really, the gills are really, you know, floating out there, kind of erect, and, they're ventilated by the head movements and by the currents, or by body movements too. In tadpoles, the gills are internal, they're inside the opercular chamber. And that's ventilated. The water that's flowing over the gills is by what we call the buccopharyngeal pump. And we'll talk about that a little bit later. But basically, you're pumping a fluid, you know, through the mouth and across the gills. And then there are gills in embryos of direct developing and viviparous species. And there's a figure in your textbook that that shows that. I don't have that here. And then some adults, for example, the sirens, remember those? They have external gills. What's that? Can you think of another adult salamander that has that has external gills?
The axolotl.
Axolotl would be one, yeah. And the mud puppy, Necturus. That's the proteid that we have there.
Oh, yeah, the human fish too, the ulm,
What's that?
the human fish, the ulm, Proteus sanguinis.
Yeah, yeah. Okay, oh, and well, untransformed, neotenic, Idaho giant salamanders would be like that too. They can't, I guess an adult should be whether it's reproductive or not,
Yeah.
not whether it's transformed or not. Then the skin, is real important in amphibians, and that's because they have that smooth, moist, and relatively thin skin. Now, for a reptile with a keratinous, you know, covering and the scales and the thicker skin, that's not appropriate really for much gas exchange. But for amphibians, some of them use their skin quite a bit. This is probably this is a great example here. This is a hellbender. It's the same as those big guys over there, and that really greatly increases the surface area of their skin. Other examples, there's really quite a few of them. I probably should have put this in another place because it doesn't really show the skin part, but this was an article that I found in the New York Times a number of years ago. This is a lungless frog that they found in Borneo. It had no airway opening in its throat, only a membrane lining its thoracic cavity. Probably absorbs oxygen through the skin. And like it's in these cold, swift flowing streams,
So it
like a
doesn't-
hellbender.
So it doesn't have a trachea? So it doesn't have a air opening in its throat?
In its throat, no, there's not something going into the thoracic cavity because there's no lung. Oh, this is an interesting speculation. Negative buoyancy, it makes it easier to sink and stay put rather than being swept away in fast moving water. So that would be, you know, that could be a really strong selective pressure for losing your lungs if you're in that kind of situation. Some aquatic amphibians, yeah, we've said that. So this is the one called the Lake Titicaca frog. Later on, we're gonna look at a movie by, called Toadskin Spell. It's a BBC nature film, and they have some really neat videos of of these Titicaca frogs that are doing push-ups like in the water, and they do that to ventilate. And they've got really, yeah, you can see how extensive the skin folds are. These guys are in trouble. One of my students, former students, went down there and looked for them, and he said, while they were there, they couldn't find any at all. And then this is, what? Trichobatrachus robustus. It's a called a hairy frog. And they have these filaments that come out of the skin. And they're highly vascularized, and so they increase the surface area. And there's a number of hypotheses of why that is the case. I don't know of any experimental studies that have been done, but the males, you know how the Ruskin newts, those males during breeding season get the real thin skin and they get the big tail, you know, that allows them to stay underwater longer to court the females. And then these apparently guard the eggs. They have parental care and the male stays with the eggs. And so this will help him remain with the eggs. Male European newts develop crests and tail fins, and the Ruskin newt does, at least gets the tail fin. Okay, so those are all things to increase surface area for having more ability to exchange gases. Tadpoles rely heavily on cutaneous gas exchange, but reptiles not so much. They have a dry skin, there's a layer of lipids there, so there's, and scales and thicker skin, and so they have little cutaneous gas exchange. There are exceptions to that. Apparently, these kind of sternid turtles, Sternothrus, you keyed out one of those, they have that ability to exchange some across their skin. Acrocortis, in 2 weeks, or wait, I guess, actually, this won't be till next month that we talk about these guys. But this is the file snake. It's a primitive group of snakes from Australia. And they just spend most of their time in water, and they can exchange gases across their skin to some extent. And sea snakes, they can dump the majority of or a lot of the carbon dioxide across the skin. And then there is some carbon dioxide exchange that occurs between the scales of things like boas and the lacertid lizards. So where there isn't the scale, there's that thinner area between the scale, and that there could be some gas exchange there. The next non-pulmonary gas exchange surface that we'll talk about is the buccal cavity, the mouth cavity, basically, and the throat, the pharynx. So as you know, plethodonid salamanders don't have lungs, and so they use largely their skin and then the buccal cavity. Softshell turtles have filamentous projections. They have projections into the throat that are heavily vascularized, and they can spend more time underwater because they can pick up oxygen through, basically ventilating their these projections in the throat. And I've heard that they could get 20–30% of their oxygen requirements that way. This was a really interesting one. These are, there's two species of pleurodire turtles. What are the pleurodire turtles again? What distinguishes pleurodires from cryptodires?
Was it,
Their neck into their.
Yeah, the neck. So the pleurodires are the side neck turtles, and the cryptodires are the hidden neck turtles, because you really don't see as much of the neck. Anyway, these guys use cloacal bursae. Now, in the past, I've lectured gas exchange across the cloaca of cryptodire turtles. In the cloaca, I had lectured, what I had learned and what I had read was that takes place in the cloaca of some cryptid like painted turtles. And then last week I read an article. It was an article. It wasn't really, it was in something like Smithsonian Magazine or something like that. It wasn't an actual research article. But they were contesting that, and they kind of reviewed the literature and said that although cryptid can have non-pulmonary gas exchange, they think it's more important that it's coming across the mouth. And they think that the actual evidence for cloacal exchanges is not very strong. It is, though, for the plurideers. So this is one of the side neck turtles from uh, Australia, and they pump water. See if I get this. They have these bursae that come off the cloaca, and they have these fimbria, these projections into those bursae, and they're vascularized. It sort of reminds you of those filamentous projections that would be in the soft shell turtles in the throat. And about a third of their gas exchange during dives can take over. And I'm not sure about hibernation for these guys, but it would be more than enough for hibernation. I saw a video. I've looked all over for it. I can't find it online, but I saw a video that was done of this, was showing the actual, and it was really, I mean, it wasn't subtle. You just saw this flow of water being forced out and sucked in and forced out and sucked in. I mean, it was a real active thing, like breathing, you know. I mean, it was, but they were using the cloaca. Okay, the degree of partitioning and the folding of lungs is related to the size of the animal, the metabolism of the animal, and the degree of non-pulmonary exchange. So let's talk about that. The bigger an animal is, what are you gonna see? Are the lungs gonna be more partitioned or less partitioned?
More.
More. Higher metabolic rate? More. Okay, well, higher the metabolism, the more of the oxygen needed, right? So you're gonna have a higher oxygen demand, and the way that you increase the surface area is by folding, creating folds and partitions within the lung. And then the degree of non-pulmonary exchange. So the more pulmonary exchange you have, what happens to the lung?
They increase in size.
The more the non-pulmonary gas exchange? The more you rely on like the skin or the cloaca or gills, something like that.
Increase.
What's that?
The more you rely on the other functions,
The
more
less
in size.
you'll be. And then what's the extreme case of that?
Losing the lungs.
Losing the lungs, right? Lungless salamanders, right? Good. Okay, so how do you ventilate the lungs? You need to get the air to those surfaces, or the water to those surfaces. For lungs, you need to get air there. So they have there's two basic types that we see. We see positive pressure ventilation, where you're actually pumping air into the lungs, and you have negative pressure ventilation. Negative pressure ventilation is what we do. We basically create a lower pressure in our chest, and that sucks air in. Where with the amphibians, what they're doing is they're creating a greater pressure in the mouth, and then they're forcing the air down into the lungs. So they initially air is forced into the lungs by raising the pressure in the buccal cavity. It's used by amphibians. So initially, what they do is they expand the buccal cavity, right? So that creates a negative pressure relative to outside. And then air, when they expand the buccal cavity here, then air will come in. Then they'll close this. Let's see, do I have a picture? Yeah, here we go. So this is positive pressure. By the way, that Jason Pilarski is one of the physiologists in our department. And Jessica Whitaker Fornick was a student working with him. She worked with breathing rhythms in birds. in developing birds. But she did this lecture and she came up with a number of, really good slides. So this is one that she found. So on inspiration, they pull the floor of the mouth down. That increases the size of the buccal cavity. And then that'll basically, air will come out of the lung and also come into the buccal cavity. And then when the buccal cavity contracts, so, when you raise the floor of the mouth there, that will force some of this air out and some of the air down into the lungs. There is some mixing, so this is not a perfectly efficient process. But our process isn't perfectly efficient either, is it? Because we have a lot of dead space when we're inhaling and exhaling. There's a lot of that air that's moving that's not in contact with an exchange surface. What animals have really efficient gas exchange? What vertebrate? Birds. Yeah, birds is a flow through, kind of goes like that. It goes in like essentially a circle like that. So the air always passes over a gas exchange surface. It doesn't get stuck, and part of the way in there.
Don't crocodilians breathe like that?
Yes, yes, we'll get to that. That's great. That's not something that's widely known. In fact, that was just discovered, I think, this century. So, I just throw this in there because it's not really gas exchange, but it's something that, chuckwallas, you keyed out one of these. They're an iguanid lizard down in the southwest, and they actually use buccal pumping to fill up their lungs. And then they wedge themselves into these cracks, and then the predators or humans can't get them out of there.
They're like the pufferfish of the lizard world.
Yeah, yeah. I'm not gonna take you to these, at least this link used to work. This was a woman's lab at, I guess, Brown University, but she had videos of all these different herps breathing mechanics. So if you're interested in this, you could go where you could look at that. So then on the other side here, we have negative pressure ventilation or aspiration. And this is actually from a human sort of type diagram, but it works, sort of works for reptiles. You have the chest. Now this has a diaphragm. What's the only reptile or reptile group that has a diaphragm?
Crocodilian.
Crocodilians, right. The others are using their costal muscles. They don't have that partition. But and the turtles are doing something else, and we'll talk about that in a minute. But basically, rather than changing the pressure in the buccal cavity, they're changing pressure in the chest, okay? So if you contract the diaphragm or some of the costal muscles, that is going to increase the volume in the chest and then decrease the pressure. And then so air will rush, move into into the chest. Then if you relax some of those intercostal muscles and relax the diaphragm, and when you relax the diaphragm, then it, bends up like that, then that decreases the pressure in the chest, and then air will be forced out of the lungs. Okay. So, and this is what reptiles use. Respiration is triphasic in tuataras, lizards, and snakes. They have exhalation, inhalation, and relaxation. By the way, since I put the PowerPoint in there, That does have all the notes, all the extra notes under the, if you're interested in that. Okay, now there is sometimes in locomotion, you're going to have conflicts between ventilation and locomotion. You know, when you watch lizards and snakes too, but in lizards, they have this side to side, you know, kind of sinusoidal movement of the vertebral column. So this is like a monitor lizard, I think, here. That's moving. And with that, then that interferes, you know, that's compressing and expanding the lungs. So varanids can take kind of incorporate that into their breathing cycle. And I think they also use sometimes buccal pressure in the buccal cavity to influence that too. Anyway, this is tied to the fact that a lot of times you'll see animals move intermittently. You see this a lot in invertebrates, where they'll move and stop and then move and stop and move and stop. And that may be related to and in lizards, for example, that may be related to this conflicts with negative pressure breathing. With the way they move the body can influence the pressure in the lungs. Let's see, in snakes, the left lung is reduced or gone, and the lung can be really long. It can extend quite a way. I think like in the sea snakes, the lung extends way down most of the way of the body. And the lung has two parts. It has an anterior portion, which is vascularized, and it's partitioned. It has sort of a honeycomb appearance. And then a non-vascular part that's that's called the air sac toward the back. And apparently, now, and I talked to a physiologist from Duke, and they had done some experiments with snakes where they had them breathe radioactive dust particles, and they were able to do X-ray videography or X-ray film back then, I think it was. And what they thought was that they were seeing unidirectional flow. But I've never seen that in the literature. However, let's see. Oh, we'll talk about turtles and then we'll get to what Ace was talking about, unidirectional flow with crocodilians. So negative pressure ventilation. Oh, one thing I should mention here too. Some of the sea snakes can do some pretty interesting tricks where I guess they can like trap oxygen in that air sac in the non-vascular part of the lung. And then they can dump carbon dioxide across the skin. And by adjusting their posture, these things dive, you know, like tens of meters underwater. And by adjusting their posture, they can get that air out of the air sac up to the vascular part of the lung. And so that they can prolong their time period that they can be underwater. And they can stay under, I think, long time. They can go deep and stay down there a long time. Now turtles, and I think I've mentioned this to you before, but here you can actually see it. So their ribs and things are fused to their carapace. So they're not gonna be able to expand or contract, you know, that shell to change the pressure in the lung. So this is what they do is they have muscles that go to the girdles. Okay, like here to the scapula and to the pelvic girdle, and here the names of these muscles aren't important. But the point is is that by contracting that and moving the limbs, they're changing the pressure in the body cavity. And that changes the pressure within the lung. And so I think if they pull, if the muscles go this way, if whatever the contraction and things of the muscles is to pull these things toward, that decreases the pressure here or increases the pressure here, and that'll cause expiration. And then the opposite of that will decrease there, and then you'll get air flowing into the lungs. So when we go this Saturday to the aquarium, they have some redfoot tortoises in their kind of jungle room in the South America exhibit. Watch those red-footed tortoises and see what they do. And if you watch them long enough, you'll usually see that they're rocking their legs. You wouldn't know, if you just look at them and don't think about it, but if you watch them for a while, you'll see that they're rocking their legs, and that's the equivalent to our chest, you know, expanding and contracting. Crocodilians, are not using their trunk muscles like these other reptiles or like the squamates are. They have the functional equivalent of a diaphragm. And so this is just like in a mammal or a bird. When that contracts, then that lowers that, decreases the pressure, and air goes in. And then abdominal muscles can be involved, and relaxation of the diaphragm can be involved in expiration. This is a study, and then there's been a group of really productive people working on this kind of thing with circulation and ventilation down at the University of Utah. That study on the monitors was done by a person from Utah. So anyway, what they found is that, you know, like birds, that they have this, you know, complicated circulation or real efficient circulation where it's unidirectional. It's flowing in one direction. Apparently, alligators do that too somehow by going to different parts of the lawn. And the implication of this, both from the bird side and from the crocodilian side, is that that's what dinosaurs had too. And dinosaurs, as you know, a lot of the dinosaur bones are really, really light. You know, they have a lot of hollow bones in them. They also have these air sacs that like go up into the neck, and some of them go out into the bones, the long bones. And that's like air sacs that you'd find in birds. And it's possible that these guys had unidirectional flow too. Okay, where are we here? Well, we might be able to actually get done with this today. So, you know, amphibians have gelatinous eggs. And so the gas exchange is mostly by diffusion, but that can be aided by convection. And we'll talk about that in just a second. Okay, so then you have different shapes. You have different environments that are eggs and laid in, and different shapes of the egg masses from, you know, like cylindrical and side or sheets. And that's related to the oxygen availability. So if you look at like bullfrog eggs, they're in warm waters that are often, you know, not very high in oxygen content. They'll be spread out over the surface in a sheet. But if you look at the ranids, like this could be you know, one of our ranid species here, it's you know, in a spherical. Some amphibians have these foam nests. that, even if, that they keep the eggs moist. This is Jessica, Whitaker Fornick. And this was out at the O'Neill Ecological Preserve, and her project was a great idea. We just couldn't technically get it to work. But it wasn't for a lack of trying. But this is a northern leopard frog egg mass that we had found. And she's got a syringe here with that green dye that the stream ecologists use, you know, to look at water flow and the streams. And she would inject it here, and then she could see that that it was being pulled into the egg mass. And the idea, the study that she had seen and was trying to replicate in the field was what had been done in the laboratory. And what you see here is they've injected ink here, and then you're seeing it travel up through these channels that are in the egg mass. And I think what's going on is that you get really big temperature differences of several degrees Celsius between this water and between this, you know, here, because these eggs transmit light, and this gets much warmer than the surrounding water. And I think it sets up these convective heat cells, and then it pulls the water through through these channels in the egg masses, increasing the gas exchange. Anyway, what would happen though is when she'd inject that, she could see that some of it was going in, but then it just, she wasn't able to visualize it adequately. Okay, and then the last thing has to do with eggs of reptiles, and that is diffusion through the cell. So in crocodile and some turtle eggs, they have real hard, you know, they have relatively hard shells. They have crystals of calcium minerals, and then there's pores that are in them. They're more like a bird egg. And then some of the other ones, the lepidosaurs and some turtles, have these filamentous or fibrous eggs, and then there's gaps between them that the oxygen can flow through. So eggshells are porous, there's gas exchange across those pores, and then it is actually goes over this chorioallantoic membrane. Okay, so here's the allantois, here's the chorion, and it's that dual membrane that the gas exchange occurs across. Hey, I finished gas exchange in one time, and I only went 1 minute over.
Chuck, did you by chance get to email the Gatorland yet?
No, did you send me a request to do that, or did I ask you to do that?
You asked me, I did not.
Okay, yeah, send it to me and then I'll start it, and then it's on my list of to-dos.
So I just email you and